Welcome back to my channel. In this lesson, we're going to be continuing on with passive processes used to transport molecules in and out of cells. In the last lesson, we looked at diffusion. In this lesson, we're going to be looking at osmosis, which, as I said in that session, is a special form of diffusion. So what are we going to be doing in this lesson? We're going to be talking about what osmosis is, defining it and describing it. Then we're going to talk about the definitions of keywords like solutes, solvents, and solutions. We're going to look at the concept of water potential and describing how that works. We're going to talk about partially permeable membranes. And then we're going to also talk about how water potential is measured. So let's have a look at some of those key terms that I was talking about. So what is a solute? A solute is a substance or molecule which dissolves in a solvent. An example of that would be something like salt in salt water. Or maybe sugar in a sugary drink, and so on. Next, we're going to look at the concept of a solvent, or what a solvent is. So solvents are the substances in which solutes dissolve to form a solution. An example of that would be water in salty water. So next question, or next word we're going to be looking at is solution. So what is a solution? Basically a combination of a solute and a solvent. So this is the combination of a solute and a solvent. For example, salty water. Okay, so that's what a solution is. So the next key word we're going to be looking at is water potential. So what is water potential? Water potential is a measure of the capacity of a solution to lose water. Okay, and it is measured in kilopascals. So pure water has a very high, or in fact the highest water potential. of 0. Anything that has, or any solution that's made up of water with anything else dissolved within it, is going to have a water potential that is lower than 0, so a negative figure. So that salty water has a lower water potential than pure water. So that means a figure lower than 0, e.g. -1 kilopascal. Okay. So on this page, we're going to look at how osmosis actually works in practice. So in that diagram, you can see that you've got two solutions on either side of a membrane. In the diagram, you've got water molecules labeled, and the water molecules are the small light blue circles. And then you've got a solute, say for instance salt, and that's the green circle. Okay, so on the left hand side in the diagram, you've got more of the water molecules, the blue molecules, and you've got fewer green molecules. That means that you've got a lower concentration of the solute, the green molecules, which therefore means you have a higher concentration of free water molecules. In essence, you have a higher water potential on the left than on the right. And for that reason, you end up with those water molecules, the light blue circles, being able to cross from the left-hand side to the right-hand side. So I would like you to think about how you could possibly define osmosis using the terms water potential and partially permeable membrane. You can pause for a second to think about it. Now that you've had a chance to think about it, let's work out what the definition would be. So osmosis is the net movement of molecules, for example, water molecules, from an area of high concentration, in this case, high water potential, to an area of low concentration, in this case, a low water potential, across a partially permeable membrane. So what is a partially permeable membrane? So you can see in the diagram there that you've got the membrane in a sort of pink color in between both solutions. So let's try to describe what that partially permeable membrane is. This is a membrane or barrier with pores within it, allowing molecules which are small enough to pass through. And that could be something like a plasma membrane. Okay, so partially permeable membranes have pores in them which allow small molecules to pass through, for example, water molecules, which are quite small. Okay, next to that, you can see a QR code, which will be a link to a useful YouTube video, which you can watch in your own time, taking you through the same sort of concept. So let's have a look at how water potential is measured. So in this next diagram, you can see we start off with pure water at the top. Pure water has no solutes dissolved within it, so it has the highest water potential of 0 kilopascals. If you have a dilute solution of, say, salt water, then you have a small amount of solute dissolved within it, and you have a relatively low water potential, so minus 10 kilopascals. And if you concentrate that solution even further, if you have more salt within it, then you end up with a large amount of the solute dissolved, and so you have an even lower water potential, so for instance, minus 500 kilopascals. So that is how water potential works. But what is the effect of that? Let's have a look. So down here at the bottom of the page, you've got some more diagrams there. So you've got a plant cell. So here you've got a plant cell with its cell wall around it. And here you have an animal cell without a cell wall. Okay, so when you put those cells in pure water, the cytoplasm within them will have a lower water potential than the surrounding pure water, and as such, water will tend to move into the cells. In a plant cell, the effect will be a higher amount of pressure against the cell wall, making it turgid, and the cell wall prevents the cell from bursting. Whereas in an animal cell, there isn't a cell wall, and as such, it will absorb more and more water until it becomes hemolyzed, until it basically pops from too much pressure on the cell membrane, and there's nothing to stop it from basically bursting. Now let's look at what happens on the other side when you put those cells in sugary or salty solution with a low water potential. You end up with water leaving the cells. In a plant cell, as you can see at the bottom, eventually the cytoplasm shrinks away from the cell wall. And if enough of the cytoplasm has shrunk away from the cell wall, it will not be able to basically repair itself, and at that point the plant will lose turgor and will become flaccid and eventually die. So we describe that as being plasmolyzed. In an animal cell, it will lose water and shrink and become wrinkled. So you can see that there. Okay, so that's basically it on this topic. So now we've had a look at both forms of passive processes. We started off at the start of this with diffusion. And remember, we said that because diffusion only requires the kinetic energy of the molecules themselves and no extra energy, we describe it as a passive process. And as such, it will happen on its own without any external energy being given to it. And you can see there an example of that, the dyed sugar cube and how it spreads through the water. Then we talked about what the definition of diffusion is, and then we gave some examples and what actually brings about diffusion. And then we talked about different factors that can affect the rate of diffusion. So things like the difference between the concentration on both sides of a membrane, for instance, or between two different solutions. The thickness of the diffusion surface, basically how far the molecules have to travel. And we talked about the surface area of the diffusion surface. The higher the surface area to volume ratio, the greater the rate of diffusion. And we talked about how all of those can be put together in something called Fick's law to give you that relationship there. And then we talked about some examples of substances that can diffuse across membranes. And we talked about facilitated diffusion. So in some instances, because the molecules are too large to pass through pores in the membranes, they can be allowed or can be facilitated to cross the membrane with the use of protein channels within the membrane. And then on to what we've done in this video. So let's have a quick recap of what we've done so far. So here's our summary of osmosis. So from the point of view of the movement of water, it's the net movement of water molecules from an area of high water potential to an area of low water potential through a partially permeable membrane. Water potential is a measure of the capacity of a solution to lose water molecules. Pure water has a water potential of 0 kilopascals, but any solution that has any solutes dissolved within it will have a lower water potential than that, so they'll become more negative. We talked about some key words that you need to know the definitions of. So what is a solute? It's a substance which is dissolved in a solvent, for instance, salt in salty water. What is a solvent? It's the substance in which the solute is dissolved, for instance, water in salty water. And what is a solution? A solution is a combination of the solute and the solvent, for example, salty water itself. And then we talked about partially permeable membranes. And as I said just before I looked back at diffusion, a partially permeable membrane is a thin porous barrier between two solutions. So for instance, a plasma membrane within a cell. So it enables small molecules to pass through from one solution to the next. So that's what osmosis is. The main part of this that some people sometimes struggle with is the concept of water potential. And I would suggest that you do some past questions just to see how questions are asked on this topic at GCSE. Okay, so if you have any comments or any suggestions as to how these videos could be improved, or if you just need some further help or quite focused questions on what we've done in this video, please leave them down below in the comments section under this video. If you found this useful, please like the video and subscribe to the channel, and that way you'll get notified whenever I put on more videos. Thanks for watching.